My name is Hoffman. I'm a research biologist at the USEJ lab here. I'm on the Great Lakes Science team. Just to give you a sense of what that facility is about. Our research group is actually spread between two locations, Duluth and then Rosehill, Michigan. But those two facilities, in total we have about 7 scientists whose job it is to support EPA research on the Great Lakes. And that's we have more. We have 7. And what even though our home base is Duluth, our charge is actually Great Lakes Water for the agency. One of the big areas of concern over since I arrived at this facility in 2006 has been invasive species. And a lot of it's leading up to Michelle's research. And when I think about this talk and how we're going to put it together, what I actually wanted to do was, instead of sort of throwing out a lot of facts about invasive species and some of the environmental problems associated with them, and we can discuss those, certainly. I actually wanted to come back to some basic ideas in ecology that structure our understanding of invasive species. Because that not only guides the principles that Michelle's utilizing in her research and explains to a large degree why you're doing what you're doing with us, we also want to try to be sort of a fundamental way for you to connect with the information. Sort of a mental picture of how we think about the problem, because that really gives you a good footing to think about how to bring that knowledge forward to the classroom, and also gives you a footing to think about how you might study some of these problems in your own setting. So there's not a lot of slides here. This is going to be some really, I want to lay down some very fundamental concepts, and I'm going to take my time to do so. So this is a casual thing. We've got the lights off and the screen on. But stop if you have a question. Let's talk. And there's going to be a fun break in the middle. I brought the party. We're going to have a party. It's going to be great. And so I'm always just full of surprises. And so that's what we're going to do. So I want to talk about 3 things today. First, I just really briefly want to talk about this idea. What does it mean to work with a scientist? I know some of you in this room, like many of you in this room, have a great deal of experience kind of developing relationships with scientists. Some of you are at different kind of points in your career. And I want to talk about how that might work and some things to think about. I also want to talk to a greater degree about what an ecological niche is. How do we understand it? How do we measure it? And then in the context of what we're doing with this group and on the ship, why does it matter for invasive species? That's the idea. So why do you want to hang around with scientists? You're crazy enough to be here, so you must have thought that didn't sound like the worst idea ever. So congratulations. Here's how I think about it as a scientist. I'm really excited to acclimate you to our science culture, however weird it might seem from the outside. When I think about our culture, I think about our culture as a culture of inquiry, hypothesis testing. And in that, what we see is, on a daily basis, quite literally, is an interplay between data and theory. And so much of the field work we do is not just about collecting data, it's about relating it back to theory. And that's why I wanted to spend more time today talking about sort of the knowledge, the theory, than the data, because we're going to get lots of time to do that on the road. But that is not a common culture shared across a lot of disciplines. It's not often utilized in the classroom, but when it is. And so I just want to get you sort of comfortable with how we see the world. I also like to think that to some extent this provides you with some more scientific street cred with your students. When you can Skype them from one of our research vessels, when you can bring pictures of you out there working shoulder to shoulder with the scientists, that's not necessarily impress them, but say, hey, we're serious about this. This is important stuff. I'm taking my time. This is an important thing about it. I love that. We benefit too. So it's not just a two-way street. It's not just a one-way street. It's a two-way street. The scientists benefit. We get to share our knowledge, which is fun. But we also get to work a lot on our communication skills. We learn about how to communicate to different types of audiences, different levels of learners, different kinds of learners. That's really important for us, for scientists. We get very comfortable speaking to other scientists in what is essentially a technical language. And that's nice, it's comfortable, but it's not always what society needs. And of course, there is a reward for us too, and that we get to work with a broad group of people to bring the knowledge out of the world. So that's exciting for us. For you, I think the trick is to engage. I think it really is that simple. Scientists are people too. And if you engage us like people, we usually respond positively. And in that context, don't be afraid to ask questions. Shoot us emails. It's totally fine. There's nothing wrong with it. Ask us questions in the middle of the research groups. Ask me questions in the middle of this talk. Don't worry about how it sounds or if it sounds technical enough or you don't sure you have the right word. Just engage. Be fearless. No worries. It works out. In that context, too, you have to understand that resources are limited. You understand in the classroom for us, too. And this is one of the things I think that is sort of an interesting aspect of this dynamic. Cindy and I've had many conversations about this. Despite all of the interest in the public, and to some extent expressed through federal policy and state policy days, in STEM education and learning, I have to say that is largely still confined in many ways to a K-12 world. And that for a lot of us working in the field, we get not much encouragement to work with educators. We don't get professional encouragement. We don't get back slaps and high fives for it. So the people who are going to be in front of you, who are you're going to be seeing in these kinds of contexts, they must be there for another reason. I think it's important to be cognizant of. Me, I'm here, and I already told you I'm here. I love working with teachers. I'm passionate about bringing that up in the world. I want to spend my time to do so. I think it's very important. But that's something to be aware of, is that this isn't necessarily their first priority in the way that their job requires instruction. And that's fair. And invite scientists into your classroom. Any way you can. They'll come. If you pester them enough, you plan with them, treat them respectfully, you can get scientists in your classroom. With digital technology, that becomes easier. You can have respect from the lab. We have a new microscope. I can actually give you a website where you can control it from your classroom. So I can bring something from Lake Superior. I can take it to my lab. I can send you the link. Your class can actually interface with my microscope in my lab and look at what I'm looking at. So increasingly, inviting the scientists into the classroom doesn't necessarily mean my foot is in your classroom. It could be other technology. When we were on the last Coastie cruise, we had a satellite uplink. We actually did a satellite uplink with a classroom at the Shut Aquarium in Chicago. And had a teacher face-to-face. We were in the middle of Lake Superior. They were in a classroom in Chicago, and it worked out pretty well, actually. So invite them in. There's so many different ways now to get people involved. Any questions about that? Thoughts, comments? Cynthia, do you want to chime in? I know you have
I guess
fantastic experience with the science.
when I'm working with mentors, I try to be respectful and careful not to overuse them. And I just was telling him the story that Dave Mount, who's at EPLab, is a wonderful man and has given me so many resources. And I didn't want to bother him again, and so I emailed someone else who emailed him. So, and a lot of times if I have mentors, I try not to have them help more than one person because it's just, it's just too much. And then let him rest for a while before I ask him again. I guess that's what I would. They are usually more than gracious to help and give you all kinds of good information and make connections with kids, but it's a lot of extra work to ask.
The National Coastie group did a survey of scientists, and it really is true that a lot of scientists are pretty uncomfortable about going into a classroom. So they just don't know if they can relate to the kids. So that's one of the things you can do is make that easier for them by helping them know how to talk that.
Do you think there's more involvement in this area though because of things like CGRAD and COSI?
Yeah, I hope so.
I think it seems to be.
I would want to add that you can always test your grad students because
one of the things I've done with some of the scientists I've brought into my classroom is prep them a little bit, because you're right, there's some of them are not good speakers and very uncomfortable. And one of the things that I got them to do is tell stories. Don't get into the technical blah, blah, blah. Just tell us a story that you think is funny. And sometimes this dry humor is just, firing. It's
Feel like
just,
magic, bro.
it's just wonderful, But it's great.
I would add to that the profs at UMD. I went through UMD at my student taught in Carlton. I would ask some of my profs for like extra invertebrates and stuff because I had no budget. And everything that they, once they were done, I mean, they helped me with a lot of stuff, and that was part because I had helped with it. But besides just grad students, I think that profs so smart and are scientists on their own, and so they're a great resource for you guys that are around here. Use UMD. It's incredible.
The professors there, they love helping, and they have graduate students at their fingertips at all times. And at any moment, they'll be like, oh yeah, I just figured it. And they'll just volunteer their graduate students to do something. I have a friend who's like, my advisor made me go do this. Can you believe that? I'm like, well, that's cool, that sounds great. But she's not happy about it. She has other stuff. But I think it's a great opportunity. It looks great on a resume for graduate students also to help them. So, contact people in UMD for sure.
So we're gonna keep moving, but yeah, I think that's a great start of this conversation. I think recognizing people in the room who've done this, as two, as somebody you can ask, I want to try this, what would you recommend? And involving your own network into this is an important piece of that. So let's get to the sort of the topic here. What the heck's an ecological niche and what are ecological niche? So ecological niche is actually one of the most fundamental and original ideas in ecology. And it really frames how we understand how organisms interact with their environment. And of course, there are themes of this going back into the 1800s, but the term was actually first coined in 1917 by Joe Grinnell. And the idea was very simple. It was a study on birds in California. He was a naturalist. And just thought that niche could be essentially the place an animal occupies. So it's basically the habitat requirements. Cover, how much brush this bird needs in, what was the temperature. So the sort of the simple habitat parameters around it. And then, this was the key part, it wasn't just where it occurred, but the ones that allow a species to thrive. And by that, there were two concepts. One is it had to be able to persist there, so it would be there over time, just not temporarily. And the other one is it had to be able to reproduce there. So the population would have to be able to sustain itself. That was a very simple idea of it. That was our first idea of it. 10 years later, in a paper by Elton, the idea was expanded a bit, this more broadly to include sort of the role the species plays in the community. And I think that the big advance here was the idea of we're going to include biotic interactions. I think it's sort of intuitive that predator-prey interactions are part of how we can understand animals who can do, and that other types of interactions, who you might be competing with for food, mutualisms who might help you get along, just survive in an environment that was important. And that became sort of this broader expanded role of niche. George Evelyn Hutchinson, the prominent limnologist, in the 50s was giving a talk in which he coined the term, which is perhaps not the most elegant way to put it, but he coined the term n-dimensional hypervolume for niche. What do you mean by that? Well, it's a volume, but we can't, it's in more than 3 dimensions. So it's a hypervolume because we in our brain, we can't imagine more than 3 dimensions. So we'll call it hypervolume. But none of us can do this, so we all just think about it as a sphere, like a normal volume. But we're going to use this term. N-dimensional hypervolume. And so this is a space, as a volume is, and it's going to be defined by both biotic, so these are sort of the species interactions, and abiotic, okay, that's our habitat, and again, we invoke this idea about thriving, persistence, and being able to reproduce and sustain the population. So that was interesting. We sort of rolled all these ideas into one, but this is the one I think that really got people aware. And he tried to make a distinction between a fundamental niche and a realized niche. And And this fundamental niche exists without the species interaction. So it's sort of the abstract environment, given a certain temperature or a certain water depth, a certain climate, a certain elevation, a certain soil type, some species would be able to persist there. But then the realized niche would be in the face of these additional biotic pressures, competitors, etc. And then they would be different, in different meaningful ways. And I think that's really got the ball rolling, because it made us much more aware of the sort of the interesting aspects of getting to niche. The origin for all of these theories is actually older than the theory itself, and it comes down to an idea of competitive exclusion. And if you teach physics, this might sound familiar. This has sort of the biological analog. But the simple idea is, if you have species A here in blue and species B here in yellow, and we were to map them on as niche, in sort of niche space, so to pretend that's temperature, that's cover, or soil moisture or something. The simple idea is that these two species cannot occupy the same niche simultaneously. One must outcompete the other. They both cannot persist. They occupy it exactly. Like, for pictorial reasons, it's possible, but the concept there is that those students are perfectly overlapping. That cannot happen, is this idea. Which means that one of these species must change. Or maybe both will change. They have a distinct niche. So now, in this case, B has become better at specializing in this bullet, a smaller range of, say, soil moisture and a smaller temperature range. And so within that little world, it can outcompete this generalist who persists over a broader range of environmental conditions. And that's the competitive exclusion principle. Now there's a subtlety that is drawn, I want to point out. I do these circles. Why did I draw those circles? Because it looks pretty.
It's because there's variables within the species.
Sort of, that's getting at it. Well, think about this. So normally we think about, you would say, oh, something can exist between, say, 5 and 15 degrees centigrade. If I did that, that would be a big line. And I say, well, it might exist between, let's say, an altitude of 6,000 or 12,000 feet. Well, that would be as representative. Better it's a circle. So something's going on here. I'm implying that they can't exist in the corners of that. So it's not box, it's a circle. Does that make sense? How would I draw that? So what's the implication that I've excluded the drawing? What it's saying is that if it's, well, it likes that temperature here over this range of elevation. If you put it at the edge of its range of temperature and the edge of the range of its elevation, it's got 2 points simultaneously. So both would be in the same place, same temperature or whatever various way. Yeah, extremes. Right. Right.
You guys all hear that? No, that was the answer.
Have one extreme, let's say the soil temperature is extreme, we have the altitude extreme, two, that would be too much. And one or the other. That species we have one of the extreme or the other.
That's right. So we've excluded out the extremes. Walk that back even a step farther out. And what you can understand then is that this actually denotes something about where it prefers to be. This is a functional kind of space. So that the additional pressures of two extremes are not preferred. They can't be there. It's taken on too much. And so maybe we'd actually think of it doing better in the middle. Where it can deal with the tolerance or extremes on one end and the other. So we're actually assuming in here, although I didn't draw it, I didn't even say it, we've actually applied something about fitness. There's that thriving piece. That these conditions influence fitness, and the sphere is implied that fitness is built in, that at the extremes there's a loss of fitness, and the animal can only take so many extremes at once before it is unable to reproduce and persist. Does that make sense? It could be a square. An animal could exist. I mean, there could be animals out there. It doesn't have to be a circle. but I just wanted to help you understand the graphical interpretation of this. It could be a triangle. Think about a marine species, right? What if this was salinity? This was high salinity. Well, it loves high salinity. And maybe as it gets down toward lower salinity, it becomes less tolerant of other conditions because of the salinity stress. Maybe it's a wedge. So the shapes are built in. I just passed that circle here. But I want you to understand that there's implications to that.
Be sure to ask questions, you guys.
So if I was to go plot this, it may not look like a circle. That's okay. The theory doesn't demand it's a circle. The circle is, all it says is that they can't have the same shape and the same time.
Question for Joel.
So,
Just trying to figure out how you use it. So let's say we have two species that are separated, and speciation is biological behavior or whatever. Separate, something like that together. They both seem equally well suited for the interactions and habitat adaptation.
yep.
So, still in some logical sense, one on some basis is going to help the other.
That would be
It
the
might be,
presumption.
the time scale is, we have no idea.
That's right, and that's a great point. And in fact, this forcing out from one species to something different than another can occur in the absolute present day sense. Two organisms competing out for the same space, and one having to adjust. But it also can occur over other shade, up a little more later on. Yeah. And so, in some sense, the fact that this has already happened, well, it's sort of a shadow, a ghost of competition that happened before, that has now expressed itself by the fact that I witnessed an animal behaving in a more specialist way versus the generalist. Because they adapted and changed their behavior, or even their genotype evolved in response to this. But here's the problem. It's really difficult to measure a niche. Why? Well, in some sense, I just told you. It's because the observed distribution, the thing I see in the world, reflects a compromise to maximize fitness in response to many pressures. That's why it's a sphere. The sphere is the compromise it's made to not be at the edges under certain conditions, to not be always uncomfortable, maybe sometimes uncomfortable in some ways, but not in others, still able to provide the persons. So this is the realized niche, and it's very hard to understand that fundamental niche that they actually occupied in the world. Scientists have had a long legacy of doing things to try to understand this. This is a classic example, work on intertidal rocky shores. People started moving around barnacles, and what they noticed is that if you exclude one species of barnacle, the other species of barnacle can occupy both the upper and lower intertidal range. But if you add it back in, competition occurs, and then the one barnacle is forced into only the upper intertidal. So here's your, it's a generalist here without that interaction, and all of a sudden it has to become specialized on the upper intertidal. Because we've added in a competitor that's utilizing some of the same resources, the both barnacles and both upper. Classic example. We have to manipulate the system to know that. There's also a long history of trying to use natural data, unmanipulated data, to understand its presence versus absence. That's tough because, again, all you're doing is pretending compromise. But if I can measure that under lots of different circumstances and times and places, maybe I can sort of reconstruct something close to that data. Maybe. And then a whole other area of interest over the years, and this is in some ways actually growing, especially with modern molecular genetic techniques, allows you to get a much better handle on in almost real time which genes are being passed along. What does that mean for what if we do something about tensor fitness? So what if I really knew which animals were thriving in which places? Would that tell me more? Because maybe some places I'm measuring where it is, it's the population is dying out. It's not thriving. It's sort of persistent, but it's not a sustainable population. Maybe it'll be gone. Maybe it's because of climate change. I'm not sure. But maybe that's a good representation of its good habitat. And I think especially the fitness piece, where it's an interesting one, there's a lot of emerging research in that. balloon. Okay, you cannot do what you want to do, which is blow your balloon up big. Okay, small balloon. Leave space. Because we're going to squeeze it.
The world's greatest morning.
Feeds our students. Crazy.
This is a trick.
It's like hide up.
Yes, please try it out.
Tools full of hot air. So you can feel
All right.
it matter.
Oh, the caffeine's in these.
Oh, do you want one of these?
Yeah.
Oh, shorter one, she just said.
There's always something, sorry.
Yeah.
My teacher didn't tell me not to do that.
Is that a shorter? Is that a, I think that's the right
I distinctly heard say that after class.
You got it.
Did I hear say that? I don't have any trouble like that.
Do you mind? They should find some Marshall hardware on the way here.
Are we supposed to time?
Yes, please go ahead and time. This is the best I could do on a science budget, I'm sorry.
I know how you feel.
So, more often than not, when you look at these studies, we often do represent the niche, at least idealized, as these two little spheres. So, we can imagine three different, here you have an aquatic organism, abstract aquatic organism, pick the one you like best. And we've thought about water temp, and a little bit about water quality, turbidity, and a little bit about the depth. And we sort of, all right. So this is your aquatic organism. Hard to complete, right? So remember, this is what you observe. This is the realized niche. Okay, this is the thing. This is it, your critter constrained by three different variables in three-dimensional space. That's what this is. Okay. So the thing is, we don't know what the true niche is. We don't know what the thing looks like. We just know this is. I'm sorry. Would you like me to do it for you?
Is everybody paying attention? Pay attention.
So, right. Let's imagine Aaron and Annette are actually two different places in the world. Okay, two different places. So, this is an aquatic animal. Let's say it has a fairly wide range. So let's call it here in New England. We'll call it Great Lakes. And you two, without looking at each other, squeeze on your balloon in some way to create environmental pressure in the niche in sort of a locally specific way. As much as you're willing to. All right. Now place them together. So in some abstract world, if we took the sum of these to balloons, we'd have a more complete niche. Understand it, right? So now let's say I'm the Midwest. Let's say this is like a little lake critter.
Wait, are those the same species?
The same species. These are all the same species, different places. So we have New England here, we have the Great Lakes. This is going to be lakes in the Great Plains, and it does something totally different. And if we had a big sheet of paper here, we'd outline it. It's fun. The point is that each one has a different shape, it's expressed differently. So each one's a realized niche. The sum of those gives you a bigger space. Can you see that? Now we could also do that over different times. So you can imagine that, you can separate the niches. So you can imagine that one is sort of, 5,000 years ago, and one is today, or maybe pre and post climate change. But the idea is that this is actually what we're often doing in the field, literally, is that we have all this information about realized niches. We've pushed around the animal in some way, whether it's in an experiment, by giving them a temperature trial or salinity trial, whether it's in the field by collecting presence absence data from different regions, different kinds of streams, or different kinds of environments. And we mash all of these realized niches together and hope that the result is something that looks something more like a fundamental, something broader than a fundamental. That's really often what we're doing with these kinds of image models. And so this is why it's really hard, because we don't know if we've collected enough pictures. And all of this is constrained by the rows we know, right? So I can't really, especially using data from the world, I don't know why an animal may not occur in a higher salinity than it really does in the world. And it may be that if I put it in that higher salinity, it actually does pretty well. It's actually not an uncommon result to find. You take a freshwater organism, put it in mild salinity, and they can actually do pretty well. We don't ever see it in nature. So it's actually really hard to get an understanding on the full tolerance capacity and ability to thrive of an organism. It's incredibly different. Not a trivial task. And that's sort of the point of this little party here, was to help you kind of understand that this is all we have. These are literally all these balloon species in space. We're trying to assemble them into some grander ability. So the question then is what?
Does this connect back to fundamental realize
Yes, so this is the realize.
when they're all together?
Yeah. When they're all together, what we're trying to do is we're create a fundamental. If you're a lover of Plato, you might see in these are the shadows on the wall, kind of right here. Right? So these are where we don't really get to see the true fundamental. What we see are these reflections. So don't focus on the crazy grass and grass. I'm going to cut this out neatly. So this is a version of this idea. Here are our balloons. This is a legitimate science paper published in the Proceedings of the Natural Academy of Sciences. And this is them putting their balloons together to show you that this is actually an evolutionary space. So these are different genetic subpopulations responding differently to these niche variables. So the niche axis 1 and niche axis 2. And they're mashing them up to try to figure out what this bigger thing is.
I have a question about that. So in that area, before we decided that two couldn't live in the same niche, is there overlapping there, or is there not overlapping?
That's a good question. So these are supposed to be evolutionary, so they are overlapping in these requirements, but presumably they're genetically isolated. There's also physical isolation.
Okay.
So how do you show that it
That's
So
really.
Joel, when possible, repeat the question.
Oh yeah. I'm sorry. So the question is, what if this sort of evolved eventually into a new species? That's a really good question. You could actually add that new species to this. Symbolically, it would be these are representing the same. So what you might draw now is a dash. I don't know, a really old heavy one. You could add one, and what you would expect to see is again some displacements there. Again, that idea that exclusion it cannot apply the same spirit. So that was 100 years of trying to understand why animals persist in space in 20 minutes with balloons. But the point is that tools have gotten better over time, our observation skills have gotten better over time, higher resolution, temporally and spatially, better kinds of informational fitness, such as genetics. But we're still, all of this is responding to some of these same fundamental ideas. One of the modern applications of this idea is invasive species. So we talked a little about biodiversity. For those who are interested in more biodiversity, we're happy to sort of bring a larger discussion of biodiversity and invasive species issues to another piece of this workshop. Fully happy to do that. We don't want to spend time here today, but I just want to recognize that invasive species today represent one of our major global biodiversity. It is one of the most obvious human imprints on the planet, is that we've systematically moved around species. And where they go, to a large extent, there are often unpredicted impacts, including things like local species extinctions, human health threats, other impacts to human activities, and the management runs globally into the billions and everything. One big example of that Sea lamprey control. Sea lamprey were accidentally introduced unintentionally in the Great Lakes by the creation of the shipping canal that allows Duluth to be an optimal port for the rest of the world. In doing so, the sea lamprey invaded, they crashed the native predator population, lake trout. Today, the U.S. alone will spend over $20 million a year on lamprey control. Just in the U.S., just a year. Canadians spend the same amount. Very expensive, this global lamprey. Why do we do that? Why are we aware of this? Ecological and also social impacts loom large. Niche theory can help. That's what we want. We'd really like to know where these things are going to go next. Niche theory can help. And so that's what I want to kind of wrap up on, is this idea that some of the most basic information we want to address these global problems come from these very basic ideas. How does that work? So, I want to just kind of really narrow down two topics here. One, there's this idea of open niches. What's an open niche? That's a strange idea. An open niche is the idea that if you go into an environment, there may be habitats or combinations of places and conditions where there aren't many animals naturally. They provide a natural opening. In other words, a place where you don't have to compete. So if the idea begins with competitive exclusion, we get this differentiation because of competitive exclusion, then this is the flip side of that. If there's not much competition, you might have an opportunity to just come on in because there's nothing to exclude from you. And much of our understanding of why and where invasive species goes is predicated on this idea. The species are going to end up, they're more likely invaded, there aren't natural competitors there. Round Goby in the harbor here is a great example. Round Goby, we've been here for many years, decades now. We don't really have a large group of fish that live on the bottom eating small things off the bottom. There weren't many here naturally. We're a relatively young system, 10,000 years old. That niche, no species had evolved to fill that niche in a very dominant way, and Round Goby just walked around an open niche. Very looking for competition. The other thing is that once established, these non-native species have been dominant in the ecosystem, lacking a natural competitor. So, if the thing that constrained you to be a specialist in where you came from, and you get into a novel environment, and that isn't there, you may respond by expanding along any one of your many environmental axes. Because you could have done that, right? So remember, beef was a generalist too, originally. So it has the capacity for that. It had just been kind of hemmed in by competitive interactions. Released from that, it can take on a whole different role. And so the question then is, well, what happens in a novel environment for species B if species A is no longer present? Go back to our balloon story. If I measured this over and over again in its native range, how would I possibly know this is true? How could I predict? This is all I ever knew. So these are actually, the implication is that these things are also very hard to predict based on available data and even with the best observation systems, because those observations are constrained by the series of interactions that define it in the modern system. Very difficult. What's the result? Well, you know some of the stories. There's kudzu, right? The Japanese ornamental that has taken over the southeast U.S. Under climate change models, that plant is in southern Wisconsin within 50 years. Another one, Corbicular fluminea, we have this one out here in Blue Spirit Harbor. It hasn't taken off like this here. This is a shot from a river down in Florida. I've been on some of these rivers with Corbicular invasions. It's incredible. They're literally carpet the bottom. Parts of the top. Carpet plant. So, no native clam-eating predators. The plant just takes it. You know these stories. There's lots of them out there.
So how does this work?
Michelle can talk to you more about this on a quick on this. But here's the basic idea. If I know where an animal comes from, then I can take lots of available environmental data and pile it all on and say, okay, these are all lots of things that might limit its ability to go into these other things. That might explain why it's in this range. And so I'm going to actually compile all that, and scientifically I'm going to develop a model by doing what's called a subset technique. I'm not going to use all that data. I'm going to take representative, create a distribution, which maybe represents a little bit of fitness, and take that information. And then, if I can do some statistics over this larger area to understand the context for these, I want to know them after. But the idea basically is that I can then constrain it in a way that gives it context, environmental data context. Now I have my environmental variable later from here. I have my where the species occurs from here. And now here's my mean again. From this, I can do tests. I can backcalculate and figure out which of these things are most important to defining the range, and then I can kick that out and make a prediction and say, well, here's what we think should occur. And I can do that over its current distribution. And in the context of invasive species, I can take that same data in a foreign place, a place not where that species currently is not, and ask, could it occur there? Would it be suitable based on its current distribution?
So what you have there isn't including other species though?
This is for one species.
That's only environmental variables though, not other species.
That's right. Now you could. So this is potentially limiting environment. If you knew about important species-species interactions, or maybe you could define ecologically like you need this, maybe more generalized than species X eats it, but things like that species eat it. There's other kinds of species here. That's where you could look at general rules. And it's the general rules that allow you to construct this with particular areas. I would say that most commonly it's done this way. Most people do not have a lot of biological species-based information. By and large, the way this is done today, it's largely reliant on most of the environmental data. That's right. You could exactly. So one thing could be low density of things that we produce. And you might have that as a functional food web piece as one of your variables. And if you knew a lot about, you could have lots of fish that eat shellfish in the U.S. that you could have that in. Yeah, absolutely. Well, and people do that.
But they're fairly new.
They're fairly new, and they're very difficult. Some of them have been very active, some of them less so. In fact, here's a good example. And it's less so. This is actually an EPA report. This is the habitat model. This is rough. So they said everything in red here was really good for rough. And everything the blue that's cool, that's doesn't like it. And this is just based on environmental data, just habitat. And this here is a little map, that's what rough are going to be. And I don't know. You can do the squint test, but they don't look a lot like it. And even the authors of the analysis, they say these things look a lot like it. And they want to argue that's not the point. There's lots of things that are missing. But that's one of the problems we have is that some of the basic information we have isn't enough to do a good job, to the point of good arguments. So for example, in this case, and Michelle will talk more about this, information about basic life cycle connectivity, where does it spend time, where do you want to go, and how is it? There's no fitness response. There's no evidence that maybe one is better than another. Maybe somebody's on self-sustaining populations, but they aren't particularly good. It's marginal at best, but the animal will be there. It shouldn't be in the model. And then those last pieces we talked about, species-species interactions. So things that might eat it, or things it needs to eat, or other interactions it requires to fulfill various parts of its life cycle.
So, yeah, public perception, when a model comes out, like let's say that's the EPA one that came out, 4 years ago.
Right.
How does that hurt the public perception? It's like models and prediction. Boy, it's not happening.
Right.
Where are you guys thinking?
So I think and that's a great point. So one of these things about these things as warning systems is that to do it well then requires a lot of communication with the public to understand the limitations of what we can do. I think the best example that you guys are all familiar with right now is eDNA, Novel DNA test innovation. There was a news article went nationwide 18 last week. eDNA found in the Fox River in Wisconsin, in a big lake Michigan.
Does everybody know what's this one?
No.
So this idea is that animals slough off cells and mucus into the environment, and it just floats out there. In mitochondrial DNA, it degrades really slow. And there are little pieces of mitochondrial DNA that can hang out there for a long time, days, weeks. And if those are species specific, I can just scoop up a piece of water, I can analyze it on my sequencer, I can get back those 15 or 18 species specific base pairs of mitochondrial DNA, and I can tell you, this thing shed DNA. So maybe there's an Asian company. And that's going to really help this one. So it's a huge maybe. There's this tremendous leap between I found this tiny bit of DNA that was originating in an animal and the conclusion that ergo animal is keeper. And in fact, we know there's lots of things in between. Everything from the bird ate the animal 30 miles away, pooped out animal scales there, that sloughed off DNA and you picked it up, to fishing boats, turns out they move eDNA, barges move eDNA. Lots of things move eDNA that aren't fish. And then you can just, because it's a very small assay, a very small piece of information, it's not, there's a inherent false detect rate. So some of those are just going to be false positives, just like any other diagnosis.
So is it the press that's running with it, or is it
So it's-
scientists
yeah.
going for the first line?
I don't know how to describe blame. It really puts the onus on a lot of people to behave responsibly. A lot of our media systems aren't set up well for that to happen. That's fine. I'm not saying that there aren't individuals in that environment.
I have a couple questions about invasives. So, do all invasives that come typically grow at a rapid rate or a more rapid rate than they would someplace else?
That's a great question. So in general, you know that invasives, as a big generality, tend to obtain some, well, either they don't need to sexually reproduce. So that's really easy, because these are a population. If they do sexually reproduce, they mature quickly, months or maybe a year. They tend to have fairly high reproductive capacity. They can produce lots of eggs. Zebra mussel will reproduce multiple times within a season. There's no stop. So do Asian clam. They have to draw thousands of young. There is some evidence that you can get a higher population growth rate in a novel environment, especially if there's the resources are plentiful. There's no competitors. That's part of ecological release. You get to capitalize on all those resources. Sometimes they'll put out either more young, or the young put out are healthier. The young put out, you're not putting out there to compete with them, so they survive really well rather than normally. So we often see the survivorship or reproductive rate increase, at least initially. It may over time decline as it reaches carrying capacity. Initially, it often is quite fast. So that's a very typical profile, is fast break maturity, very high reproductive capacity. Also, they're often generalists. They often survive in lots of different kinds of environment. So there are essentially lots of opportunities for different niches.
So are they linked genetically to each other necessarily or not?
Yes, but you can still see founding effects. And so the question is, are they linked genetically? There's a whole field of study actually understanding how these things are linked over big spaces, continents, based on genetic founding effects. So if you did, say, let's say a ship was over in the Caspian Sea a week ago and dropped off some group here, the group here inevitably would most likely have a smaller genetic pool than in the native range. And depending on how the genetics were structured in the native range, we will even tell you, oh, this is the Caspian Sea group, because their genetic profile is unique to that area and we match it. And we can actually see a re-invasion of that group. So there is a genetic link. It's actually, we can measure it and study it. And it may have implications for how the animal survives. It's going back to the evolution of why. Different subpopulations may have some different level tolerances. So maybe the one that you got has some interest in a different level tolerance. It may be positive or negative.
Now, one other question about that open niche that you were talking
Yeah.
about. Does that necessarily, is that a healthy environment or not healthy? Or doesn't it matter?
It's a great question. Could be either, depends on the animal. So, in the severe case, so John Linger in Ecological, he's the DNR fisheries biologist who was leading a bunch of the restoration work on the harbor. John was excellent about how the fact that we didn't have any invasives in the water quality control. Basically, the river was so unhealthy that nothing could persist. So it was sort of a natural protective against things going to fly ships. So there's an element where if the environment is so degraded, it's probably not going to get much information. But there's probably an interim rate where it's probably partially degraded in general, maybe not as there. It's used to be lost as an information. So they could be, but we don't have to know. It doesn't have to be.
So what would you say about openness? Well, what have we learned in terms of what happened with smelt? That seems to be bigger than the $20 million that we're spending on Africa.
So that's a great point. And so one of the, there's actually a really neat paper on this, this just came out this year by University of Wisconsin Madison. It turns out that if you look big picture at invasives, even things that so we call something invasive that basically can achieve a high level of abundance, and if it tends to cause a nuisance. Nuisance with ecological, economic, social. Turns out that a lot of these invasives, their abundance varies widely across a big part of the world. So you go to one lake and it's not that big a deal. You go to the next lake over and it's a problem. And so these niches, these open niches express themselves on lots of different scales. And it could be the scale of water. We said a curriculum. We don't have curriculum like a mad. We have a few individuals hanging out by the tree. It's our Asian with the mad plains I showed you. That animal lived here. It doesn't look like that here. But in all the rivers on this continent, it looks like a man. So,
Bill, were you specifically asking about smell as an example?
That would seem like
it smells one of those.
they're not one of those other,
In some
they have expensive controls.
yeah.
They just see the, this is
Yeah.
uneducated.
Yeah.
They just establish their balance.
Things where you're in Lake Superior, that's true. We haven't had to control them. They didn't become a problem. But our inland lakes was our decimating the local fisheries. And there's aggressive control. So the state of Wisconsin spent a lot of time and energy to figure out around this small So just to wrap up, so what's the niche good for? As a fun idea, it's quite powerful. Understanding range limits, over space, how animals interact with other animals, and how communities then respond because all those species interactions add up to an ecological community. They're really hard to characterize, and they may not be the same everywhere you measure them. It's all different rules. But it's still a very useful idea, very powerful construct, and in the context of invasive species, this concept can be really important as part of our understanding of global invasion ecology, even this kind of